we're done. And we're done. So now all the tRNAs are all attached and ready to go. Remember, there are 4 things that we need to have to get this to work. And these 4 things are our tRNA that's ready to go. That tRNA also has its amino acid, so there's another thing that's needed. We have to have our mRNA, and then our one big, most important piece, I would say, well, perhaps the most important piece, would be this ribosome. The ribosome comes into 2 pieces. It comes into a small piece and a large piece, or subunit. And the mRNA is actually read right in between those 2 pieces in very specific spaces. There are 3 places on the ribosome. Pre-AP, you don't need to know what those 3 spaces are, but you do need to understand that those 3 spots, those 3 places, are where the codon resides, where the codon is set and read on this workbench that reads in groups of 3. So how does this work? Well, the mRNA then comes in and attaches to the small subunit, and it goes all the way to this P site where it's looking for a particular sequence. What it's looking for is something that tells it to start the process of reading. So we need to get the place ready to start. Now AUG, which was the bases here, this codon here, AUG, is the actual start codon. So A, U, and G here is the start codon, but it does line up, or does set up for a particular transfer RNA. So we've got our AUG in the right spot. The transfer RNA that has the anticodon, UAG, there it is, that all lines up. It has its methionine, it's already charged on the end. AUG says start, this is where we're going to start, and so everything is ready for this particular piece of mRNA to be read. So AUG all ready to go. Now the ribosome has in its spot here AUA. Remember, we read from 5' to 3'. So we're looking for a transfer RNA that has its amino acid that's going to have UAU on it. And we do have one of those. Here it is. And bam, it comes in right in and it just sticks into that one little spot. Now notice that the amino acids now are very, very close together. We've got this red one here that has isoleucine on it, this tRNA that's lined up with its codon here, and it's attached, is very close, puts this methionine and isoleucine very close together at this A site. This P site here, where the green one is, what's going to end up happening, because they're so close, is the amino acid is going to come off of the top of that tRNA, and it's going to form a peptide bond with this neighboring mRNA, or excuse me, neighboring amino acid. And I want you to notice something here. We've got a hydroxyl and we have a hydrogen ion. These 2 things coming together can form water. And previously we've talked about the fact that when 2 small subunits come together and water is formed, a larger unit is made. That particular reaction is called a dehydration synthesis or condensation reaction. So here we go. Here's our hydrogen in our hydroxyls, and that's going to disappear, basically be formed, and we're going to have a bond form here, and let me pick that up to do that. So there's our bond. That peptide bond is now formed between these 2 amino acids. Water's been released, so everything now shifts. We've had this bond made. Everything now shifts down 1 codon, so that we now still have our AUC here, our anticodon with this codon AUG. We've got our UAU anticodon on this tRNA attached here to the AUA, and we now have our 3 here, our 3 bases here. I'm just going to, well, that won't work. There we go. GGA is lined up in this A site to be put in. Before that happens, remember that this tRNA here got rid of or let go of its amino acid so that it could bond with this next one right next to it. It's no longer needed. It needs to actually go back out to the cytoplasm and get recharged, pick up another methionine. It may be later on that we have methionine again. So this is going to go back out to the cytoplasm. So now we have the same setup we had just a minute ago. We've got our spot here, this middle spot, the P site, and we need to find, or the tRNA that's got the anticodon to GGA. The anticodon would be CCU, and so that tRNA that has its amino acid and has the anticodon will come in and set up. And again, now we have our amino acids right next to each other. This particular tRNA is going to let go of its amino acid. It's actually got two attached to it. And this particular amino acid then, this glycine that's on the new piece, comes in. Remember, it's a condensation reaction. Water is going to be removed. Notice the OH and the H are going to disappear. Here we go. And there they go. They disappear. We form a peptide bond. So a bigger molecule is made. Water is released. This is a condensation reaction or dehydration synthesis. And again, you want to push your little molecule, your growing peptide, polypeptide, your protein, you want it to be pushed up and out because we're making this molecule, making this protein, and we want that to get out and leave the ribosome so it can be used. The whole thing is going to shift down 3 again. It's going to shift down 3. Okay, so we still have our U and A bonding and everything bonding up correctly. This all shifts down together. So here our anticodon and our codon are all lined up. This molecule here, this tRNA, does not have its amino acid on it anymore because it gave it to the growing chain. So this now has to leave. We're set back up again. We've already read these first couple of, first 3 here of your codons. We now have a new codon in, and I didn't quite make it quite right. It should all fit in that nice little piece there, but it's CCC, our new that's our new codon that needs to be read. Our anticodon then would be GGG, and we have that. This is the proline piece here. It's popped off again. There we go. And this GGG anticodon on this tRNA is going to come in. And again, we have our amino acids very close to each other. This growing amino acid chain gives up, or it comes off of the tRNA. We've got our hydrogen ion, our hydroxide of the carboxylic acid, and our hydrogen here. That's going to make water. Remember, anytime a bigger molecule is made and water is formed, that's a condensation or a dehydration synthesis reaction. So we're going to put that together. The bond is called a peptide bond. Each of the amino acids are called peptides. We have this growing chain of amino acids going up and out of the ribosome. The whole process, this whole thing shifts down. So it all shifts down. One codon. And now we have our GUA here ready to go. And that's our codon. But before that can be read, this particular tRNA has to go out and find its amino acid again. So it's going to go back out the cytoplasm and pick up its amino acid. Here's our codon now, GUA. So we need to have CAU, and we have that. There is a tRNA that'll come in, and it's got its amino acid attached to it. It comes in. There we go. And again, it's so close here that we're going to form that bond with this growing chain, but before that happens, this tRNA has to come off. So here's our growing chain. Here's our valine with its particular piece on it. The OH group is here, and we're going to come in, get rid of a water, attach it with our, and we'll have a peptide bond. This reaction is a condensation reaction or dehydration synthesis. We're all set and ready to go. The growing amino acid sequence, the growing protein is going up and out. The whole thing shifts down again, down one codon. So there we go. We have our new codon here in the A site ready to be read. We've got this tRNA that no longer has its amino acid on it, has to go back out to the cytoplasm to pick up another amino acid. And so now we need to read the CAC codon. Its anticodon would be GUG. And so CAC, just to reinforce a minute ago, we had our amino acid chart, and CAC is histidine, so we're looking for a tRNA that has the right anticodon with histidine attached. and here it is. Our anticodon would be GUG, and there it is, and it all lines up. Again, the amino acid here and this one are so close together that they're going to bond together so that this tRNA gets rid of the chain. Amino acids, excuse me, the amino acids here and this amino acid join together. The hydroxide comes off, or is removed, and the hydrogen on the amine group is removed. It may be kind of hard to see because these are light colors, but it gets removed. So water comes out. Condensation reaction or dehydration synthesis, peptide bond forms. We now have this growing protein growing out of the ribosome in a big long strand. The whole thing shifts down one codon there, a set of 3, so it's all shifted down. Our last one here on this chain then is UGA. Now we had a start, a thing that said start, and on our codon chart there's actually 3 things that say stop, and one of them is UGA. So we look at this codon here, UGA. Well, that says to stop. There are 2 others here, UAA and UAG. Any of these would tell the protein, or tell, excuse me, the ribosome to stop. And what happens is a special protein comes in that has the anticodon in it, ACU, to that UGA, and it's called a release factor. Now in pre-AP you don't have to know any of this, but it just makes sense to show you, so you have an idea of what's going on. There's something that has to tell it to stop. And what it does is it just comes in and says, hey, we're done, and all of this stuff now breaks apart or comes apart. The mRNA comes apart, the tRNAs go apart, this disconnects, this comes apart, the ribosome, the large and the small subunit also come apart, and so that really all that's left is this protein, this sequence that's got its individual peptide bonds here between them, all formed in this process called translation. And you're done. If you'll notice, there were 6 amino acids together, and to form that, there were 5 condensation reactions that occurred, 5 peptide bonds that were formed. This finishes up our model on protein synthesis.